Hello, everyone. It is my pleasure to introduce a multi-centered prospective birth cohort study in Western China. Prenatal and early childhood development periods are critical for human lifelong health, where diverse determinants from maternal nutrition to environmental exposures can impact fetal growth, birth outcomes, and future health risks. However, understanding the complex factors influencing prenatal and postnatal development is challenging. Various potential internal and external determinants and the long latency between early exposures and later life health outcomes. Birth cohorts, by longitudinally tracking individuals from the earliest stages of life, provide comprehensive insights into the progression of health and disease over a lifetime. Globally, several national birth cohort studies such as the Danish birth cohort, UK birth cohort, Norwegian birth cohort, Finnish birth cohort, Australian birth cohort, Italian birth cohort, Dutch birth cohort, and Singapore birth cohort, have advanced our understanding of early life health determinants. In China, birth cohort research began in 1993 with studies on folic acid and neural tube defects prevention. The China birth cohort study later recruited participants across 17 provinces to investigate birth defects but ended follow-up at birth. Other cohorts have focused on economically developed regions in eastern and southern China, leaving western areas underrepresented. The China Southwest birth cohort included parts of the Western China, but the enrollment was limited to Sichuan, Chongqing, and Guizhou. Western China, which comprises approximately 70% of the country's landmass with nearly one-third of its population, presents unique health challenges due to geographical, environmental, and socioeconomic diversity. Especially, the high-altitude environment with hypobaric hypoxia and the diverse ethnic populations with distinct genetic backgrounds, cultures, and dietary habits, contribute to variations in maternal child health outcomes. To address these gaps, we launched the WCBC cohort, a multi-center, longitudinal and large-scale prospective study. The enrollment was conducted across 8 clinical centers in 5 provinces in Western China, including Qinghai, Gansu, Guizhou, Chongqing, and Sichuan. The cohort included 9 follow-up visits, from early pregnancy to 6 years of childhood. A total of 12 different types of biological samples have been collected. WCBC also collected the maternal medical record, delivery records, offspring medical records, maternal questionnaires, and offspring questionnaires based on the follow-up schedules. By integrating advanced multi-omics measurement, including genome, proteome, exosome, metabolome, spatial transcriptomics, single-cell RNA-seq, culturome, metagenome, and virosome analysis, WCBC provides a valuable platform to explore gene-environment interplay, early life determinants of health, and long-term disease risks in diverse populations in Western China. Participants were distributed across high altitude, like Qinghai and Gansu, middle altitude, like Guizhou, and low altitude areas, like Sichuan and Chongqing. More than 76% of the participants were of Han ethnicity, with 19.3% belonging to minority ethnic groups. The top 10 ethnic minorities included Hui, Tujia, Tibetan, Miao, Dongxiang, Tu, Yi, Zhuang, Bai, and Galao. Women from high-altitude areas conceived earlier than those from middle and low-altitude areas, such as Gansu and Qinghai. Most of the participating women had given birth, and their children had reached 2 to 3 years of age. In general, prenatal follow-ups were conducted at 10 to 16 and 32 to 38 weeks of gestation. Offspring follow-ups have been conducted from 1 month to 3 years of age. WCBC had collected more than 200,000 biological samples, 220,000 medical records and 80,000 questionnaires. WCBC is a large-scale, multi-center, prospective study enrolling over 15,000 pregnant women across 5 provinces with diverse altitudes and ethnic populations in Western China. It enables comprehensive investigation of altitude-related, ethnicity-specific, and socioeconomic determinants of maternal child health outcomes. It integrates longitudinal data with multi-omics to advance understanding of early life determinants of health and diseases and inform public health strategies in Western China.